Hello, my dear listeners. In this session, we want to look at classification of hormones. In the previous videos, we have looked at different endocrine glands in the body. We have continued to see how hypothalamus controls the anterior. We have seen different axes. And in this session, we want to look at classification and the mechanism of action of hormones. Please, if you haven't subscribed, remember to subscribe, share your comments, and like the video. And under classification of hormones, hormones are classified in two ways. They're classified in two ways, whereby we have classification according to the chemical nature, what we call chemical classification, and also we have classification according to function, whereby according to chemical nature, we shall have hormones classified as protein hormones, or you can call them peptide hormones. We shall see hormones being classified as steroidal hormones, and lastly, being classified as amine hormones. Amine hormones meaning they are delivered from amino acids. So these are the major three classifications under chemical, whereby we have protein hormones, steroidal hormones, and amine hormones. Protein hormones, for them, as their name suggests, that they are made up of peptide bonds. They're made up of a polypeptide chain, and of course this polypeptide chain is made up of several amino acids. And these hormones, they are water soluble. And because they are water soluble, they have their receptors on the surface. Their receptors are found on the cell surface. Because we know water-soluble substances do not cross a phospholipid bilayer. So they have their receptors on the surface. And these hormones include all hormones produced by P glands. It includes hormones produced from P glands. Any gland that begins with letter P, that is pancreas, pituitary, parathyroid, there is pancreas already. So all hormones produced by, there is pineal. So you see, even God made life easy, that because they begin with a P, they are also produced by P glands. So all hormones which are produced by pancreas, parathyroid hormone, pituitary, and pineal gland, they are all water-soluble, or what we call protein hormones. Whereby in pituitary we saw them, there are many LH, FSH, TSH, growth hormones. In pancreas we saw insulin, we saw glucagon, we saw somatostatin, all of this. In the posterior we saw oxytocin and antidiuretic. All of those are protein hormones. Apart from that, we see steroidal. And these steroidal hormones, for them, they are derived from, you can call them, these steroidal hormones are fat-soluble hormones. They are fat-soluble. And these fat-soluble hormones, they are derived from cholesterol. So when they ask in an exam that which of the following do we derive steroid hormones, is that they are derived from cholesterol. And these steroid hormones are produced from gonads and adrenal gland. We find them in the gonads and adrenal, and specifically adrenal cortex, whereby gonads we had ovary and testis. producing androgens. That was estrogen and testosterone. Testosterone, dihydrosterone, then adrenal cortex also producing small amount of estrogen, but first it produces glucocorticoids, which we call the cortisol, cortisone. Then we saw mineralocorticoids, which we saw aldosterone, and still smaller portion of estrogen and testo in a smaller amount. And these hormones are known as steroidal, derived from cholesterol, which is a fat. And because we are saying they are fat-soluble hormones, for them, their receptors, these hormones, they have their receptors within the cell. Within the cell. It can be either in cytoplasm or cytosol or in the nucleus. So, and we see their major mechanism, which as we shall see they are responsible for protein synthesis, or we can see them in activating enzymes. So these are steroids. The last group under chemical is amine hormones. Or these amine hormones are derived from amino acids. And amino acids, we have like amino acid tyrosine, amino acid. We have tryptophan, amino acid. Whereby tyrosine amino acid is the one that is used for biosynthesis of T4 and T3. Whereas tryptophan is used for catecholamines. And these catecholamines, we saw epinephrine, norepinephrine, and dopamine. And these ones, for them, their receptors are found either on the surface or inside. For example, T3 and T4 being smaller in size, their receptor is found in the nucleus. Because they are smaller in size, so they can cross the phospholipid bilayer. But these catecholamines, some of them have the receptors on the surface or in the cytoplasm, depending on their molecular size. So this is the classification of chemical classification of hormones, whereby we have protein, steroidal, and amino derivative hormones. And I've seen proteins being produced from don't forget this, produced because it starts with the P, so it is found in all P glands. So whenever a gland that begins with the P produces protein hormones. And we have said they are water soluble, therefore they have receptors on the cell. And steroidal being fat soluble, their receptors are found within the cell, and they are found in the glands and adrenal cortex. And not forgetting that they are derived from cholesterol. That is very important. And then these ones are derived from amino acids, whereby it is tyrosine. Tyrosine is for T4 and T3. Then tryptophan is for catecholamines. Then lastly, about the classification is classification by function, whereby classification by function, hormones are divided into either tropic hormones or direct effectors. And tropic hormones, these are hormones, this one refers to hormones of anterior pituitary gland. Now, these hormones of anterior pituitary gland and sometimes hypothalamus, they have ability to cause hyperplasia of the cells they are triggering. That whenever they are overproduced, they cause increase in the size of the target site. And whenever they reach at the target site, these tropic hormones, they stimulate the target site to release another hormone. And it is this hormone that will exert the final feedback. But which is different from direct effectors. That for them, when they are produced, they directly act. And they do not cause hyperplasia of the target site. An example of this hormone, of the tropic hormones from anterior, this one includes all hypothalamic hormones are tropic. Or hypothalamic. Then from anterior pituitary, we have guys like FSH, LH, have TSH, and we have TSH, FSH, LH, and ACTH. So these four are tropic hormones. Why are they called tropic hormones? Is that once they reach on their target site, they stimulate them to produce more enzyme, more hormones. And with these hormones that will exert their final feedback. So they are stimulating other glands to produce more other hormones, which is different from direct effectors. This is the word is direct. Effectors like growth hormone and prolactin. We see for them, when prolactin goes to the mammary glands, it directly stimulates production. And it does not cause hyperplasia. We see growth hormone, when it goes to the tissues, it causes directly growth without triggering another production of the hormone. So this is according to function. So basically this is classification. And according to solubility, which is number 3, they can either be water soluble or fat soluble. And we have seen steroidal or fat soluble, then we have seen protein hormones as water soluble. So that is basically classification. Then I want to end by talking about mechanism of action. The mechanism of action of these hormones. So mechanism of action, and I want to begin with water soluble. Water soluble hormones, which we said that these water soluble hormones have their receptors on the surface. So if I can draw a cell, this is a cell. These ones have their receptors on the surface, and they are connected to the G-coupled receptor, which ends with adenyl cyclase enzyme, which is always inactive without an enzyme. Then here we have a nucleus, and this nucleus has DNA. This is the nucleus of the cell. So for water-soluble hormones, what we want to say is that when the water-soluble hormone like protein hormones come and bind on the G-coupled receptor, this is the G protein. When they bind, this is the hormone binding. It causes what we call hormone receptor complex. And it is this hormone receptor complex that causes activation of adenyl cyclase. And when we go further in the biochemistry, we shall see how GDP plays a big role. But we see adenyl cyclase being activated. What activates adenyl cyclase is that hormone receptor complex causes the conformation change in the G-coupled receptor. And when it causes conformation change, and this G-coupled receptor has 3 subunits, whereby it is divided into 3 subunits. That is alpha, we have the beta subunit, we have the gamma, and the alpha. And this alpha, when there is no hormone, is always bound to GDP. So when the hormone binds to form a hormone receptor complex, GTP is released from the alpha subunit, and we see GTP coming to bind. GTP binds. So GTP comes and displaces the GDP from the alpha subunit. And then after this, we form alpha GTP subunit. And it is alpha GTP subunit that activates adenyl cyclase. So we are seeing this complex that when there is no hormone, GDP is bound to alpha. But when the hormone binds to form a hormone receptor complex, there is a conformation change in the G-coupled receptor, causing GTP displacing GDP from the alpha subunit, forming alpha GTP complex. And it is this alpha GTP complex that acts as a lock to activate adenyl cyclase. So when I have the alpha GTP activating adenyl cyclase enzyme, so when it activates it, we see this adenyl cyclase going to hydrolyze ATP to cyclic AMP, what we call CAMP. And this hydrolysis reaction of active cyclase, which is activated by alpha GTP subunit, is the one that increases CAMP level in blood, in the cells. And when cyclic CAMP, which is known as a second messenger, this is a second messenger, is going to, in the cell cytoplasm, they are protein kinase type A, which are always inactive, and they are activated by increased cyclic AMP levels. So when there is increase in cyclic AMP, which is a second messenger, it goes and also activates protein kinase type A. And whenever these protein kinase type A are activated, the one that goes to the proteins in the cell, if I have proteins in the cell, it goes there, it goes to the cells and starts phosphorylating by adding a phosphate group. And when it adds the phosphate group, these proteins can be enzymes, and these enzymes become activated. The enzymes become activated. What is activating them is the phosphorylation by addition of a phosphate group. So when a phosphate group is added by protein kinase type A, and it adds to a protein like an enzyme, it becomes active, and it is the one that exerts the final work of the hormone. So these guys, they work through a second messenger mechanism, whereby we have said the hormone receptor, when the hormone binds, it displaces the GTP displaces GDP from alpha subunit, forming alpha GTP complex, which comes and activates adenyl cyclase. And it's this adenyl cyclase that is activated that hydrolyzes ATP to cyclic AMP, which is acting as a second messenger. The hormone was the first messenger. Then after increased levels of cyclic AMP activates the inactive protein kinase type A, which have phosphate groups. And this, it comes to the enzyme and starts adding phosphate by phosphorylation method, causing them to become active and exerting the effect of the hormone. So this is the mechanism of action of water-soluble hormones, which is different from the mechanism of action for fat-soluble, or what we call steroidal. For steroidal, if this is the cell, we said for them their receptors are found inside the cell, and if we have a nucleus with DNA, for them their receptors are found within. So we see their receptors inside the cell. It can be in the nucleus or in the cytoplasm. It can be in the nucleus or in the cytoplasm. And whenever we have these hormones, if this is a steroidal hormone, for it, because it is a fat soluble, it crosses the phospholipid bilayer. And after crossing, it can either come and bind in the receptors in the cytoplasm, or it can bind to its receptor in the nucleus. This is the nucleus. And when it binds, it is going to cause this DNA to go under replication. And when it replicates, it forms a copy. It replicates and forms a copy by transcription method. And this copy is known as mRNA. They form mRNA, and this mRNA is smaller in size. It can move via nuclear power and it goes into the cell where there are ribosomes. And these ribosomes are going to act as machinery. But if I bring mRNA here, sorry, this mRNA it undergoes translation to form protein. And it is this protein that is exerting the final mechanism. So we have seen that for steroidal and smaller molecular size hormones like T4 and T3, their receptors being in the nucleus, when they bind to form receptor hormone complex, causes replication, whereby we are unwinding the DNA strand, and these strands of DNA, they undergo transcription to form mRNA. And this mRNA, or what we call messenger RNA, exits via the nuclear power, goes to the ribosomes in the cytoplasm, and these ribosomes act as machinery for synthesis of protein by the process known as translation. And this is what we call the central dogma of the cell, and whereby we can say hormones act by forming genes. mRNA is a gene, and this gene we can use it to synthesize new proteins which can exert a final feedback. For example, like steroidal hormones, which we need to grow. So those are the two major mechanisms. One by activating enzymes. The second is by synthesizing new proteins. And the third one is by causing cellular permeability. I can write them here. The final is either they can act by activating enzymes in the cell, or they can act by forming genes, that is mRNA, that can be used to synthesize new proteins. Used to synthesize new proteins. And then lastly is that they can do by altering permeability of the cell. So these are the major three outcomes when these mechanisms come into play. When either we use surface receptor or intracellular receptors. The end result is either activating enzymes which exert the effect, or forming a gene via central dogma, forming new proteins which exert the major effect, or alteration of the permeability. Thank you so much for listening until the end. Always make sure to subscribe, always make sure to share to others, and may God bless you.